morning, friends. It's a wonderful Wednesday morning. Here we are, just loving life in Ottawa, enjoying a little dusting of snow. You can't go wrong with that. Anyway, we don't need to dwell on the weather anymore, my friends. Let's get right into the lecture today. We're talking about the genetics of language and personality. And you might be asking yourself why it would be that these two topics are combined. The answer is really that we're running out of time in the course and there's too many things that I want to talk about. So at a certain point we have to begin merging topics in order so that I can say something to you about a variety of things. So here we're kind of taking a bit of a grab bag approach and putting language and personality together. They're both, I think you could say, psychological dimensions of normal human life. Everyone has a language and everyone has a personality, and in both cases you might have pathological states, but even if you don't, you have some, you're somewhere on those continua. So we can put it into that bin if you like. But mainly the reason that they're together is just because they're two interesting things, and we can fit them in, or we can give a reasonably good survey of both of these things without going too far overboard. So here is the lecture slide, the introductory slide, and we'll begin with language. So imagine if you're like a Martian or a space alien or something like that, and you're observing life on Earth using whatever tools you have available. Most likely what you would notice, if you have some kind of deep level of Martian insight, is that life on Earth evolved on the basis of a highly conserved universal language known as DNA. I don't know how exactly you would discover this, but it took humans, many tens of thousands of years to figure out what DNA was. But we're assuming that somebody capable of, interplanetary travel would figure this out much more quickly. And so it's true that every living thing on Earth uses essentially the same language of DNA. Codons and genes are, for the most part, universally compatible. So it is possible to take a human gene and put it into bacteria. They can read that gene just fine. And if you were so inclined, you could do the reverse. And the genetic language is essentially the same for everybody. Now, the contrast here is that animals don't have any type of universal communication system. DNA is, in a sense, a language that communicates genetic information between generations, but animals, and of course DNA relates to kind of the evolutionarily derived hierarchical nature of all life on this earth, but language doesn't fit that criteria. Language is not universal in the slightest. No animals share a communication system, or no different species, I should say, particularly share a communication system. And what's more, even humans at least have different languages that they speak amongst each other. So there's quite a contrast there, an incredible diversity of communication systems. If you think about the difference between, say honeybees communicating by dancing, to birds communicating by bird songs, to whales communicating by those sort of whale vocalizations, and other kinds of creatures communicating on the basis of pheromones, chemical signals, not even physical auditory ones. There's quite a lot of diversity there. And the language, as far as it goes, is a capacity that's unique to human beings. Other animals have, very or relatively sophisticated communication systems, but it's debatable as to even whether the fanciest of these really constitutes a language in the way that humans have it. It's, human language is recursive, meaning that you can nest individual components together. You could sort of say, like, I went to class, learned about language, and then went for a coffee. And you've nested 3 separate clauses together into a sentence that would make sense to anybody who heard it. And even your fanciest animal communication system isn't capable of that level of complexity. And that level of, or that ability to create infinite numbers of new combinations of words and phrases. So it's a relatively unique, it's a capacity unique to human beings. And that kind of makes for an interesting genetic question, because most of the things that we have looked at in this class, while humans are unique in a certain way, we do share some overlap with other species. So for example, if you're looking at the genetics of neuroanatomy, well, neuroanatomy in the human being is different from other animals, but they at least have brains, and they have all the normal parts that we do. We just have different versions of it, and so on and so forth. So language is a rather unique capacity. So the fundamental question you have to answer is what has gone on with our genetics that has given us the capacity to learn language. And language is, like many aspects of your phenotype, it has to be acquired during critical windows of early development. And another important point is that language and cognition interact. So it's a little bit difficult to tell where one begins and one ends. How much of your thought process is really just you going through sort of a linguistic process in your own mind, kind of what somebody like B.F. Skinner would have called covert speech, or maybe John B. Watson. But back in the olden days, behaviorists used to think that there was no thought per se; it was just that you were talking to yourself, which is an interesting kind of thought. But anyway, the two processes do seem rather intertwined, and of course, humans are unique in their high capacity for both. Now, does language have a genetic basis? In a trivial sense, it must, because human beings have the unique capacity to use language, and what differs between humans and our nearest living relatives is about maybe 1% of our DNA. Chimpanzees are almost the exact same as us genetically, but humans are different in certain key ways, and whatever those differences happen to be, they're sufficient to create a human who's capable of speaking and learning languages instead of a chimpanzee who's capable of neither. Many neurodevelopmental disorders include language delays. And so right off the bat, you can start to think that to the extent that these neurodevelopmental disorders are genetic or have a genetic etiology, there may be some overlap between that and the genetics of language. So for example, ASD, autism spectrum disorders, we see an association with a delay and or limited vocabulary and expressiveness. It's not exactly a language disorder per se. It's considered to be sort of a neurodevelopmental disorder. However, the effects of language, or the effects on language rather, are sort of baked into it, as it were. Language delay can also appear on its own. This is perhaps more relevant to our discussion. If you have language delay in the presence of other sorts of cognitive deficits, well, then it's difficult to say whether it's a delay specific to language or if the whole brain just is not able to operate on a high enough level to use language. It's a bit like asking if your computer can run a piece of software. Well, maybe it can't because that particular software uniquely doesn't work. So, if you're trying to install an app that isn't supported by your phone or whatever. Or it could be the case that your phone could run the app, but it's just too bogged down by other processes and it doesn't have sufficient resources, whether it's memory or CPU, to actually run, say, the YouTube app, if that makes any sense. So when language delay appears on its own, that tends to be that tends to be especially interesting from the point of view of the genetics of language. The previous terms for this would have been things like developmental aphasia or developmental dysphagia, meaning like a lack of speech or problems with speech. These terms are no longer favored because aphasia to a neurologist means that there's a neurological problem that's interfering with language. So if somebody may be in the late stages of, say, Alzheimer's disease and eventually lose their ability to speak coherently, and this would be called aphasia. And we don't tend to like to use those words for these developmental conditions anymore, because that implies we know what's going on in the brain, which oftentimes we don't. We prefer the term language disorder. And according to the DSM, this is nothing more than saying that the individual has a consistently hard time using language in different manners, whether it's speaking, writing, or sign language or other. Sign language, by the way, despite the fact that it doesn't involve your vocal cords and the normal kind of mechanics of language, other than that, behaves very much like language. For example, babies who are born deaf, they won't babble vocally so much as they'll babble with their hands using kind of a primitive form of sign language. So it's rather interesting how your brain's tendency to want to develop language is so strong that even if you don't have the normal apparatus for hearing and speaking, your brain will figure out some other kind of solution to the problem. And it'll be there waiting to learn whatever language-like thing there happens to be. If you learn sign language instead of, say, spoken language, that's just fine. Your brain can work with that just as well. which is kind of interesting, I guess, because it decouples the question of language from the anatomy of the vocal cords and so forth. Obviously, being able to speak using your voice is convenient in many ways because you don't, it's an organ made specifically for that purpose, but it's not the only option. But anyway, you see things like reduced vocabulary, limited sentence structure, or a limited ability to put words together to form, grammatically correct sentences. The grammatical things are especially interesting, I think, because it implies that there's some problem with how the brain is organizing the sequence of words, which is rather interesting, especially if the person is cognitively otherwise pretty solid. Now, does this language delay have a genetic basis? Well, it comes in many forms, so we don't have time to investigate every possible thing. You have problems with the sequencing of words. Others would have to do with the choice of vocabulary. The child might know many words, but may put them in the wrong order. Put them in ungrammatical orders. Oftentimes children will do this anyway, but they'll be quickly kind of corrected and begin to use the proper grammar without too much trouble. But if children persistently use sort of unconventional grammatical forms, that's the potential sign of a language delay. Now, the sequence of events in a language delay is fairly consistent across the variety of conditions, and of course also between people, but there's a wide variation in the rate of development. So some people have delays that appear very quickly, other people have delays that appear rather late. The rate of language gain is quite variable. Some people are early talkers, some people are late talkers, even within the normal spectrum of human variation. Then, of course, when you get into the pathological section, some people are a little bit late, other people are very, very late, other people never acquire language at all. So there's quite a lot of variability there. And the genetic contribution to this seems to be sort of proportional to how severe the deficits are. So in a twin study back in 1998, and there are many, many studies like this is just one example, Dale and company found that severe cases of language delay, the bottom 5% of people, in terms of their vocabulary, vocabulary means to name or to call in Latin, which comes from a Proto-Indo-European word meaning to speak. So rather interesting. But anyway, so those severe language delays at age 2 are 73% heritable. That's what this chart is indicating here. So what they're showing here is for each of these different bins or categories of people and their vocabulary status, whether you have 5% of the normal vocabulary or 100% of the normal vocabulary for your age, the relative genetic contribution to this, determined by twin studies, is variable. So people who have the most impaired vocabulary have a very large contribution from twins. In other words, monozygotic twins are highly concordant, and a comparatively small influence from the environment. But by the time you get to what we'd call sort of a normal or an age-appropriate level of vocabulary, the concordance between monozygotic twins drops off to below 30%, and the shared environment begins to have a very large impact. As it should, really, if you think about it, when you're talking about people, twins especially, who are age 2 and below, there's not a whole lot of non-shared environment in that scenario, right? How much non-shared environment, how often are 2-year-olds going off and doing their own thing privately for hours and hours at a time? Typically, you would try to make you try to combine everything into one trip if you can. You're not going to the grocery store 2 separate times with each twin. You're going at the same time. They're going to have many of the same experiences. So this is, I think, rather expected. But anyway, language delay, and severe language delay in particular, seems to be distinct from the other forms of sort of more mild language delay. So, for example, if you only have 15% of the normal vocabulary for your age, that is about as heritable or about as genetic as having the full vocabulary. So it's really the severe conditions that have the strongest genetic influence as far as we can tell. Oh, yeah, and yeah. So just remember that, yeah, the root for vocabulary comes from the same root as voice, if that connection never occurred to you. So we have this kind of awareness, I guess, that language delay is genetic, or at least inherited in some sense, on the basis of many, many twin studies and family studies. But of course, it's not something you would typically think of as following a Mendelian heritability structure. And the reason for this is that language, as you might expect, is highly polygenic. So while broad tendencies do seem to be heritable and genetic by those kind of conventional definitions, it's rare that you would ever have a pedigree where you could study what appears to be a dominant or sort of an autosomal dominant kind of inheritance patterns. If you could find one, that would be quite valuable. And in fact, they did find one. This is the KE family in the UK. So this is a family of people living in the United Kingdom, and they were famously studied by the Canadian Myrna Gopnik, who was quite famous in her own right and has a number of children who have gone on to be writers for like the New Yorker, I guess, and scientists in their own right. But Gopnik was a linguist and working out of McGill, I believe, and studied this family in detail in the UK and came to the conclusion that what the family had was a unique grammatical deficiency. The members of the family who were affected by this sort of inherited dysphagia performed significantly worse on tests that involved syntactical semantic features. So these were things like tense changing. If you were to give the person every day he kisses his nanny, somebody who wants to move this into the past tense wouldn't have a lot of trouble saying yesterday he kissed his nanny, and you would know how to change this word to, you'd change the S to a D, and you'd have a sentence that was grammatically correct. This is something that the affected members of this KE family were not very good at. And they also seem to lack a general rule for producing plurals. Normally, by the time you reach, a fairly young age, when you're learning whatever language you happen to learn, I only really know English, so we'll go with that. You acquire general rules for plurals. So Jean Berko Gleason's famous WUG test is kind of a test of this, where you try to test somebody's ability to determine plurals while decontextualizing it as much as possible. Nobody's ever heard of a WUG, so you would never be able to do this just by memory. So the test is really quite simple. You just say, okay, here's a WUG. Now there's another WUG. There are two of them. There are two blank. And the person would only have to be able to say there are two WUGs. They would have to be aware of the fact that when you want to pluralize something, you just add an S to the sentence, to the word. And of course, this, unless the word is mouse or moose. No, I guess moose you do pluralize that way, but like goose, there's all kinds of weird exceptions in English. So it's far from a general rule, but unless you hear differently, it's pretty simple to just add an S to things and wait till you get corrected if there's a problem. But it turns out that the people in the KE family really couldn't do this very well. But they weren't universally deficient. They could do other things, including instantiating reflexives. So, for example, he washes him versus he washes himself. They could work with possessives, the mother's baby versus the baby's mother. They could figure that kind of stuff out. And they could also figure out negative passives. The car is not being pulled by the truck. So there's kind of a microstructure to this type of dysphagia, at least as claimed by Gopnik, that made it seem like there was a disorder in this family that was very specific to certain aspects of language. And the fact that it was inherited in an autosomal dominant fashion really was quite a plus from that point of view. So here's the pedigree chart from this family, published from a paper in '95 when this first became a popular family to study. And so you see here we've got the grandparents, where you've got black colored shapes representing somebody who's affected by this form of dysphagia. Empty or non-filled in shapes are people who are unaffected. A cross through the person means they're dead. Circles are females, squares are males, and you can follow the family through this whole pedigree. So it turns out that from these grandparents, 4 of the 5 children were affected by this condition, yet were able to successfully reproduce on their own. And I mean, I think their spouse would be missing from this part of from the pedigree, but they would have a normal spouse with a healthy language center and so forth, and reproduce and have a number of children. And then many children would also have have this same phenotype. And just looking at it here, you can see that if somebody is a carrier, or somebody if somebody expresses this condition, the odds are approximately 50-50 that they will pass this on to their child. So for every child who's born, there's about a coin flip's chance that they'll also have this condition. This is a textbook autosomal dominant condition. And So this kind of makes it an attractive family to study, right? Because you have this concept that language is heritable. You have a family with a very specific problem in their use of language, and you have a autosomal dominant inheritance pattern for it, meaning you could theoretically localize it to a single gene that's just passed on through this family. The affected members also have lower IQ, on average 18 to 19 points below the unaffected members, which, assuming the unaffected members are average, 18 to 19 points below that is quite a severe impairment. It's not something that you'd particularly want. It's more than one standard deviation below the average as for the general population. And also problems articulating words. And in fact, there's controversy about Gopnik's claims because maybe she wanted to find that it was a grammatical disorder and not one of articulation. A disorder of articulation means that what's actually happening isn't so much that the person can't learn grammar, but more that their mouth and their vocal cords and all of the apparatus of speech can't work together to coordinate the muscle movements needed to produce words coherently. So I think the jury's still out on this. People now believe that it's not exactly a disorder strictly of grammatical rules, but one more of the articulatory apparatus and intelligence more broadly. So I'll leave that for you all to consider. There may be some bidirectionality here. I've always liked the idea that language and learning language, just like learning other tasks, requires an interaction between the physical process of producing it, the muscles and so forth, and the brain, in the same way that people who learn to play piano oftentimes just picture themselves playing piano and can almost mentally practice if they think about doing the thing a lot. So there's sort of an interaction between the process of learning it and the kind of muscle memory. Muscle memory is not real, but the quote unquote muscle memory that goes on when you actually exercise that skill. In spite of this controversy, the most interesting fact about this is indisputable on the basis of the evidence I've shown you. The language deficit, whatever it happens to be, has an autosomal dominant pattern of inheritance. And so Gopnik, of course, was there to suggest that this perhaps demonstrates the existence of a grammar gene. And considering that grammar is one of the key elements that defines human language, languages differ between population groups. There's many languages spoken in the world, but within each language group there is a fairly widely agreed upon grammar. This of course can vary by dialect, but the basis for mutual understanding of language is the grammar. And so if you use a type of grammar, if you use a grammatical form that is completely idiosyncratic and unique to you, nobody will understand what you're saying. If you use a grammatical form that other people understand, then it's fair game. There's no official grammar for English or any other language. It just happens to be what people use and what they understand. So this led, these discoveries were being made kind of in the early to mid 90s, I guess you would say, and this really put the impetus on finding this language gene. Whatever is going on with the KE family, there's clearly something interesting being inherited with them, and discovering what it is could point you toward the genetics of language. And of course, in the 90s, this was long before GWAS studies were ever being done. 1998, the big thing to do then was to watch that 70s show and Austin Powers, because I think the boomers had reached a point where they were ready to be nostalgic about the sort of the late 60s and early 70s. The boomers have now moved on, unfortunately, to posting conspiracy memes on Facebook. But anyway, the group Fisher et al. Was published a genome-wide linkage study of this family, and that's pretty easy to do because it's a pretty small family. So it's a lot easier than trying to do a genome-wide linkage study where the inheritance structure is not known. You just have to look at regions that seem to be inherited commonly with the affected people, but not present in the unaffected people. And their work suggested that the disease-associated mutation was located on the long arm of chromosome 7. Now, if you recall, with linkage studies, you're not exactly scanning the entire genome looking for every possible SNP or every possible variant. All you're really doing is you're setting up little markers on, spaced roughly evenly on all of the different chromosomes, and then you're looking for which of these markers follows the disease-causing allele. So anybody who has the disease, you look at what markers they have, and you look for the markers that people who don't have the disease don't have. And then on the basis of that, you can say, okay, well, where are those markers? And the markers will point you to a certain sort of zone on a given chromosome. It doesn't narrow it down a whole lot, but it narrows it down enough that you can kind of get to work and try to find and try to map that out in a more fine and precise way. So this was localized to the long arm of chromosome 7, and coincidentally, Lei et al. In 2001 reported the discovery of an unrelated individual named CS, who happened to have a balanced translocation at a similar location on chromosome 7, and also happened to have a lot of problems with their language. So this is one of the things that you would see in the earlier days of genetics and genomics, is this, I guess, vague awareness that a disease-causing allele was located in a certain spot on the chromosome, coupled with the coincident discovery of another person with a completely separate, inheritance, but a similar disorder that helps you localize this even better. Because now that we know there's a balanced translocation, you simply have to ask where the break point is between this translocation. So the translocation is between chromosomes 7 and 5, and the long arm of chromosome 5 to the long arm of chromosome 7. So these two pieces of the chromosomes are just switching places. And if you recall, that's what this looks like here. It's just when two chromosomes swap genetic material. And if you recall, when I talked about this last time in the disc 1 example for schizophrenia, the issue isn't so much the arrangement. You can kind of put the DNA wherever you like. It doesn't really matter a whole lot. I mean, animals across the animal kingdom sometimes have different numbers of chromosomes, but pretty similar genetic, they're pretty homologous in terms of their genetics. The chromosomes, in a way, don't matter a whole lot from that point of view. But it does matter a great deal if where a chromosome breaks is in the middle of something important, like the middle of an exon or in the middle of a gene. If you break a gene in half and then put one half on one chromosome and one half on another chromosome, that gene has become useless, because the cell will never be able to read that whole gene all at once and translate and transcribe a proper full-length mRNA. This breakpoint overlapped an already known about brain-expressed transcript called CAGH44. This was really useful mostly in the way that it helped the authors, both Fisher's group and Lei's group, to I think they're actually both part of the same group. Anyway, they allowed the authors and the people working in this field to refine their mapping of the language-associated locus. And this here just shows a fluorescent in situ hybridization image of the rearrangement. They're labeling the different parts of the chromosomes and showing how they've moved from chromosome 7 to chromosome 5, if that's something that interests you. It's funny, by the way, in the sort of early 2000s, the way you would hear the words brain-expressed transcript. That's a very odd thing to hear in the 20 years later now, in 2021. We talk about genes, and we talk about mRNAs and microRNAs and long non-coding RNAs. Brain-expressed transcript is very, very vague. But what used to happen in those days is they didn't have a genome all worked out, and they didn't have RNA-seq to go in and find all of the different, to go in and find all the different genes and how much they're expressed, and they certainly didn't have single-cell RNA-seq or anything like that. What they could do is they could just take pieces of tissue and make cDNA from it, and then pick cDNAs that were particularly abundant in one tissue and rather depleted in other tissues, and then take those and sequence them individually. And then you would have something you knew was a transcript that you knew was expressed in a given tissue. You had no clue where it was from in the genome or what it did, but you would just happen to know that it was a brain-expressed transcript. So that's a very kind of old-fashioned, quaint way of doing things, but we got to respect our history in this class. So in a different paper, they finally, glossing over some of the story here, but ultimately they use these different sources of information to properly localize where this mutation was, both in the KE family and in the unrelated CS individual. And the gene they found it's located to be, or the gene where they found this is called FOXP2. The affected individuals in the KE family had a G to A mutation. So that's what this chromatogram is showing here. This is a chromatogram from Sanger sequencing, and without getting into the details, it's just showing you the fluorescent signal from the terminator nucleotides. You see, I think when I told you about that, I implied that it was run out on a gel, but more commonly it's run through sort of a capillary electrophoresis system that does the same basic thing as a gel, but it's all automated. Anyhow, these fluorescent signals indicate that in that particular position there was a G. That's sort of a dark colored. I mean, I don't know what the fluorophore would that be, but it's represented by a black line. And then a peak from the blue signal indicates there's a cytosine in the next position, and a peak from the A signal represents there's an A, or means that there's an A in that position, and so forth. So right here, there's normally a G guanine in that exact position for the control, or sorry, for the unaffected people. They're homozygous for the G there. And for people who are affected, who have this language defect, you have a signal coming from both A, and you also have in the same exact place a signal coming from G. Now, whenever you have two signals coming from the same region in Sanger sequencing, what it means is that they're heterozygous for whatever variant there happens to be there. They've got one copy of a G allele, which is the normal healthy allele, and they've got one copy of the A allele, meaning that the A allele is the bad one, the one that you don't want. And it happens to be that this is located in a protein coding part of the gene. And what this G to A mutation does is it causes that codon to change from coding for arginine to histidine. That means the FOXP2 protein is different, and this is a loss of function mutation, meaning that for people in the KE family, the FOXP protein no longer does whatever it's supposed to do. It's a transcription factor, so some aspect of its ability to act as a transcription factor is lost. And the official term for this is FOXP2 haploinsufficiency. Haploinsufficiency just means that they only have one working copy of the protein, and following the importance of gene dosing that I told you about in numerous times, but I believe just as recently as the last lecture, you have to have the right number of copies of the gene so that it's expressed at the right level. If you have extra copies, that can be bad. But if you have missing copies, for example, if one copy doesn't work, that can also be bad. It could either be sort of a dominant negative thing where the protein actively interferes with its healthier cousin or brother, or whatever you want to call it, or it could be a situation where just maybe one of them is a dud, the other kind works, but with half as much of the working protein as you normally would have, whatever sort of cellular functions that protein is supposed to carry out don't work anymore. It could also be an imprinting thing. Maybe you've got a healthy copy, but it's imprinted and thus not expressed. That's also possible. So a lot of different things can be happening there. Now, this discovery of FOXP2 as a putative language gene set off a flurry of research, as it would if you heard that there was a language gene. It's a transcription factor, as I said, expressed in many regions of the developing brain as well as the lungs. And so, of course, they went through and made the knockout mice that are lacking FOXP2, either homozygously or heterozygously. And there are some interesting sort of, quasi-language phenotypes that they found that really raised the excitement level a fair bit. I remember when all this stuff was coming out back in the early 2000s. In 2005, I would have been just sort of starting to learn about psychology in my arts program at St. FX. But I remember hearing about this kind of stuff. They have, mice lacking FOXP2 have reduced ultrasonic vocalizations as pups. Mouse pups, mice and rats and I suppose rodents in general communicate by these ultrasonic signals. You can look up really cute videos of rats like laughing when you tickle them. Rats are very nice, cute animals. They're like little dogs. And if you have a pet rat, you can kind of tickle it on its belly and it'll sort of make these, you won't be able to hear them, but if you have a way of hearing ultrasonic signals, you can hear sort of a laughing type of noise coming from the rat. And mouse pups will communicate with their mom by the same kind of ultrasonic signal. And it turns out that mice who lack this gene, and thus, they're kind of models of the affected members of the KE family, have reduced ultrasonic vocalizations. And zebra finches, which are a type of bird, have also been made lacking FOXP2 in the basal ganglia, and they have impaired song learning. And song learning is oftentimes used as an analog for human language because the way birdsong works, it's not what you'd call a language per se, but the way it's learned, I think, reminds researchers of the way that human languages are learned. And certainly the bird's brain is primed to learn those songs, just the way that human brains are primed to learn some kind of language. So you can look at this study from Lay et al. Using in situ hybridization to localize FOXP2 in various areas of the developing brain. You'll see it expressed in the cortex, the basal ganglia, the thalamus, the inferior olives, which are located in the brainstem, and the cerebellum. We love the cerebellum in this class. We're always seeing stuff happening in the cerebellum. Half the neurons in your brain are located in your cerebellum, and could anyone say more than 2 sentences about what it does? A very mysterious kind of situation back there. Anyway, and then if you look at MRI imaging or functional MRI imaging, what you'll see is that affected members of the KE family have underactivity in the premotor cortex, which is this area here. The primary motor cortex is this area here, basal ganglia and cerebellum once again, when repeating nonsense syllables. This is a good test, by the way, that kind of takes the linguistic skills apart from the knowledge of language. The two things are kind of separate. Being able to understand, like being able to have the neural architecture that supports language is separate from knowing languages and words itself. So you can have somebody repeat a bunch of nonsense syllables like, ba 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 or whatever, and measure their brain activity and see if there's any sort of abnormalities there. And being that it's a transcription factor, you can ask about which genes FOXP2 regulates. And it turns out that it regulates the expression of a number of genes crucial to brain development. In particular, CNTNAP2, which is a cell adhesion molecule also linked to language development, one of the other big candidate genes in the language space. CNTNAP2, I believe, is the longest gene in the human genome, too. Remember, I was talking about how gene length is a thing? It's the longest one of them all, which is kind of funny because it's not a particularly long protein. It just happens to be a very, very long gene. So we have a question in the chat. Nathalie asks, with FOXP2 knockout, would the pups express a quiet laughter or would they not laugh at all? That's a very good question. If a tree falls in the forest and nobody's there to hear it, does it make a sound? If a pup laughs but you can't hear it, is the pup really laughing? I think that's a very philosophical question. I don't know how you'd, I don't know. I don't know how you'd really figure that out. I guess maybe you could sort of, if you could imagine like a human version of this where you're talking about adult human beings and you could sort of say, okay, well, maybe they can't laugh. Maybe they don't have the muscle power to laugh, or they don't have the vocal power, or whatever the case may be. You could certainly sort of scan their brain or look at other aspects of their affect and figure out whether they probably would be laughing or not. I think that's quite doable in a human. I think it's a little tough in a mouse where you can't ask them anything and you really can't scan their brain meaningfully. And I don't know. That's hard. It's, quiet laughter. Yeah, good question. I don't know. I'll think about that. You got to enjoy some quiet laughter, though, folks. If you can't laugh at yourself, what are you doing in life? Yeah, but laugh quietly. You don't want people to hear you. Anyway, so that's that. And I guess maybe as a general kind of rule for understanding the effect of different types of mutations, if you have a mutation to a transcription factor, that one mutation can have a very large effect on things because transcription factors bind to many different parts of the genome and regulate the expression of many genes. So as far as mutations go, it's a bad place to have one because not only does it affect not only does it affect itself, but it could also affect many other, or the expression rather, of many other genes, and that can cause all kinds of downstream problems. So once this was discovered in the first place, many studies were done and found other instances of FOXP2 mutation and verbal dyspraxia in families that were not related to the original KE family. So in this sense, it's rather like the DISC1 story, where you find this kind of strange family with this fairly severe phenotype that's inherited in an autosomal dominant sort of way, and then by mapping out where that gene is located, you believe, you have the idea that you found a language gene or say a schizophrenia gene or whatever the case may be, and then you look and you find other examples of it in the population. But as always, or as we've seen at least previously, this doesn't play out in the bigger picture. So in the broader genome-wide context, FOXP2 does not seem to be a general risk factor for language impairment. there are many, many different levels and degrees and varieties of language impairment and language delay. There have been genetic studies done on these, and it doesn't seem to be the case that FOXP2 is involved. Other genes are, but FOXP2 is not, and loss of function mutations to FOXP2 are comparatively rare. The reason for this is that loss of function mutations to this would be quite damaging to you and your health, so it's unlikely, as far as natural selection goes, it's unlikely that any mutations that would happen here would be favored by selection and thus passed on. So in the early 2000s, which I feel like I'm an ambassador from those days, there was a great deal of excitement about this kind of stuff because it really seemed like they discovered something. And this was buttressed by the fact that humans appeared to have a unique version of this gene. Remember that in human beings, between individuals, your status for FOXP2 can determine your risk of a language disorder or a language impairment. But then what about between species? If FOXP2 is the language gene, what about our nearest living relatives, the chimpanzees? Well, it turns out that FOXP2 is generally well conserved across vertebrates, meaning that most vertebrates have a fairly similar sequence for it. But the human version differs from the chimpanzee counterpart at 2 amino acid sites. And that is, I guess, fairly relevant because initial genetic studies of the population, or of the human population, seemed to suggest that this locus had recently undergone positive selection, meaning that this new variant, or this variant was, in the view of the earlier researchers at least, relatively new and had swept through the human population relatively quickly. So a way to think of this would be that it's so advantageous to have this human mutation, or this human version of it, that as soon as it's available, everyone who doesn't have it fails to reproduce or doesn't reproduce it successfully, and everyone who has it does really, really well. So here's the paper that first found this. This was Enard et al. In 2002. And they're simply showing the human FOXP2 amino acid sequence compared to other similar species. So you've got the human sequence up here, followed by the chimp, the gorilla, the orangutan, the rhesus macaque, and the mouse, because of course, mice aren't related to humans particularly, but we study mice quite a lot, so we have them in there too. So you have the different amino acid letter codes from the IUPAC definition, or dictionary. You have these polyglutamine stretches. Q stands for glutamine, and you've got stretches here where there's a bunch of glutamines in a row. This is a fairly common motif in proteins. And then you have two key areas where all of the animals except for humans are the same. Doesn't matter what these codes stand for, I guess, but what matters is that chimps, gorillas, orangutans, rhesus macaques, and mice all have one version of the protein here, and humans are uniquely possessing of another one. Wherever you see a dot, by the way, that just means that the species have the same sequence as us. So you can see that for most of the amino acids here, with very, very few exceptions, all of the species that they've looked at here have the same exact amino acid sequence as human beings. The only real stand-apart differences are here and here. And it was taken, or this was taken to mean that this version of FOXP2 had evolved at some point after the last common ancestor between humans and chimpanzees, which was quite a while ago, but it was, taken to be a fairly, comparatively recent thing. And if it's true that FOXP2 is essential for the production and acquisition of language, this mutation could be a really fundamental thing for what began the process that set humans apart from all of the other apes. And this is something that we're all very curious about because it's, at the same time, it seems like we're very similar to apes, but on the other hand, we're clearly very different. And it's a little bit perplexing why that is, to be quite honest. We have very similar DNA, yet we're capable of so much more than even the fanciest monkey, or the fanciest ape, I should say. And I think if I would recommend you think about that a little bit, because it's really kind of amazing what humans can do even when we don't try, compared to what apes can do even after extensive training. Apes, of course, are stronger than us. A chimpanzee could easily beat you up if he wanted to. He could do worse than that. And I guess if you think about it, we've never seen a chimp at full strength because they've never really worked out. Anyway, sorry, I'm not trying to turn this into the Joe Rogan show, but it is interesting to think about. The this kind of, I guess, belief was shaken a little bit by the discovery that this human-specific version of FOXP2 also exists in Neanderthal DNA. Now, this means that the time when this evolved had to have been at least at the time of the ancestor between for both humans and Neanderthals, which would be about 300 to 400,000 years ago. So it's not. So this simple fact is telling us that FOXP2 probably wasn't recently selected, at least not the way that people thought in the earlier part of the 2000s. Krauss et al. In 2007 were the ones who found this out, but what they're showing here is the same kind of chart I showed before. Here's the this is not the amino acid sequence. This is the the DNA sequence, but it's the same kind of idea. So you've got these two places where humans are different, and of course you can see that they're different from chimps. That's the first one. Gorillas, whatever this thing is, Pongo, and mouse. They're different from all those guys. But here in the DNA sequences of two Neanderthals that they've found, they are not different. You can see, well, here we don't have one, I guess, from this part. But anyway, the mutation seems to be absent the Neanderthals have the same version of FOXP2 as modern humans, meaning that this can't be that recent. You could also take it to suggest that Neanderthals had language abilities that were equivalent to humans. That's very possible. We don't really know, I guess. I wasn't there. So the question is, was FOXP2 recently selected? The evidence from the Neanderthal DNA seems to contract the initial supposition from Enard and company, which had suggested that the human-specific version of FOXP2 was recently selected. A selective sweep, by the way, is what they're saying it was. A selective sweep is simply when a mutation rapidly reaches fixation in a population. Fixation is when it reaches a point where everyone basically has it. And so you can imagine selective sweeps happening under very severe selective circumstances. Say, for example, there's a plague going around that only people with a certain version of the gene are resistant to. The plague will infect and kill everybody who doesn't have that gene. Thus, within a very, very short period of time, that particular gene will reach fixation in the population because the selective pressures are so strong. And I guess what they were imagining was, imagine if some people could speak and others couldn't. Anybody who could use language was at a very strong advantage to people who couldn't, because they could, plan and share ideas and all kinds of other great things. So that would have that ability would have swept through the gene pool rather quickly. And the way they had calculated this was with a statistic called Tajima's D. Tajima was a geneticist who had figured this statistic out. It's a measure of the frequency of polymorphisms relative to the expected frequency of polymorphisms if mutations are taken to be neutral. So by the estimate of Enard and company, the human-specific area of FOXP2 had a negative D value, and that just means that it was recently selected. And another way kind of to put this in a different term, or to put this in different words, a negative Tajima's D would indicate there's sort of an excess of low-frequency polymorphisms that would be relative to the expectation. This means that there's there must have been like a genetic bottleneck at some point that eventually spread out and that particular gene took over everywhere. So like again in the plague example, imagine if one gene protects you from the plague, but only a small number of people have it. Pretty soon everyone else is going to die and just those people will be left. Then their population will re-expand and suddenly everyone's going to have it. But anyway, so Enard's estimate of all this, don't worry too much about how exactly it was calculated, but the point is that their estimate was based on a fairly small number of samples. To calculate this properly, or to calculate Tajima's D properly, you need to really understand the baseline diversity of that locus. How does it differ not just between, say, white Europeans, but how does it differ from people all over the planet? And that's really what tells you whether it was recently selected or not. Human beings, despite our apparent differences in culture and geography and all the rest of it, we were all living together in Africa not that long ago as far as geological time goes. So if you take into account the wide and humans, of course, all have fairly similar capacities when it comes to language and all these other things. So if you find that the diversity between human population groups is such that your estimate of the selective pressure is thrown off, it means that it likely wasn't evolved in the last common ancestor to humans. So anyway, in 2018, finally, Atkinson and company came out and said, using a bigger and stronger estimate of genetic diversity, there's no evidence of a selective sweep. So in essence, what they're doing is taking into account people from all over the planet, broadly dichotomized as Africans and out of Africans, meaning anybody who wasn't derived from a recently African population, by putting all those people together, the evidence of selection disappears when you look at each group separately. So it's really just kind of a sign that, like, if you're using measures that depend on genetic diversity, you have to know what the actual diversity is before you'll have an accurate assumption or an accurate estimate of that measure. So this is it's kind of unfortunate, I guess, because the idea that FOXP2 is a language gene, I don't think we can exactly accept this anymore, nor can we accept that it was recently selected. Too bad. Now let's just look at the GWAS studies that we have available. We have one from 2020 by Price and Company. The GWAS studies of language ability tend to be very small compared to the ones we've seen for, say, anthropomorphic traits like body mass index or height, or even things like depression or schizophrenia. I suppose the reason for this is that nobody's willing to pay for larger ones. Language GWAS is not something that is crucially important to human health. And what's more, there are no big foundations that sort of lobby for the work the way that you would have, say, for cancer or diabetes or heart disease or Alzheimer's or these other kinds of things. So it just kind of tends to fall by the wayside, and the studies don't tend to be that large. But Price et al. Did publish one involving cohorts based in Toronto, in Philadelphia, with various levels of reading disability. And what they found was that the genes that they seem to be involved with these conditions have to do with neurodevelopment, maybe not surprisingly. So for example, you've got RGAP23. This is a gene that's involved in neuronal migration and axon pathfinding, and then also ROBO1, which is also involved in axon guidance. So you can think back to your neurodevelopment class. When your brain is, when your brain is developing, all of your neurons are born wherever they're born, in the subventricular zone or wherever it happens to be. They migrate to their location, and then they begin to extend their axons and dendrites. And the axons have these kind of growth cones that guide them to their destination. Every axon from every cell has a particular location that it's aiming for in one part of the brain, and axons can go quite a ways if you think about it. You can have axons going all the way from your primary motor cortex all the way down to your to your ventral horns in your spinal cord. You can have axons going from different parts of your cortex to other parts, and the whole guidance process is quite elaborate and quite complicated. So having problems with the wiring of your brain, perhaps not surprisingly, will lead to issues with language and many other things. So we see these axon guidance related genes and neuronal migration related genes being involved with language delay and reading disabilities and so forth, most likely because those are neurodevelopmental disorders at their root. We also see an enrichment of axon guidance related pathways in people with more general speech and language disorders. So that's the nature of language in a nutshell, is that it's not there's no one gene responsible for language. We still don't have a solid answer as to why it is humans can speak so effortlessly and chimpanzees can't. Unfortunately, we kind of lost our ability to use FOXP2 as the main reason. But within the realm of human variation, most of the genes that have to do with language ability seem to be involving neurodevelopment. So like many of the things we've looked at, language is a fundamentally neurodevelopmental process. Now let's switch gears into personality. We're going to just do a hard turn into this topic. If you aren't familiar with the psychology of personality, I'll urge you to consult this slide in detail. But in a nutshell, the way that personality is measured has been basically fixed by psychologists. They've boiled it down to what's called the 5-factor model, or what I'll sometimes call the Big Five. These are 5 dimensions of personality that seem to apply fairly well between age groups, and they apply fairly well cross-culturally, and they seem to capture most of the variation that we can measure in personality. So you've got dimensions like extroversion, agreeableness, conscientiousness, neuroticism, and openness to experience. And these images here show you kind of what the continuums are. So each of these is a continuous scale you can measure with various psychological tools. So for example, if you're high on the extroversion scale, you are sociable, fun-loving, and affectionate. If you're low on the introvert or the extroversion scale, you would be introverted, and you would be described as retiring, sober, and reserved, which I think is actually not a very kind way to speak of introverts. As a representative of that class, I think that we should be getting a nicer treatment than just being called sober and retiring. I just don't like big crowds, folks. That's all. Anyway, so high on the extroversion scale would describe an energetic and enthusiastic approach, and it includes traits such as sociability, assertiveness, confidence, and ambitiousness. Don't we love those kinds of people? Don't we just love it when people are assertive to us and sociable? Yeah, it's great. Anyway, you've also got things like agreeableness, the person's level of altruism, cooperation, willingness to conform to group norms, warmth and kindness. I think that's rather interesting. You talk about conformity. I'm sure there are personality dimensions associated with all kinds of interesting factors in our day-to-day lives, and you can kind of go down the line. But I don't want to make this into too much of a psychological class, because the last time I studied personality in school, in university, was in my third year. In third year psychology, I took a personality course, and I was actually impressed with how well organized that field is. I kind of felt when I was taking it that personality was going to be one of the fluffiest, sort of least serious topics. But to be honest, they've done great work with it, and it may actually be one of the more solid and replicable areas of psychology. So props to the personality psychologists out there. Now, personality traits are relatively stable dimensions of inter-individual differences in a whole range of things, including cognition, affect, and behavior. The most relevant thing, I think, here is that they're stable. You can be in a bad mood, or you can be in a good mood. You can be feeling sociable one day, or you could be feeling like you need a night at home with a cup of tea and a good book. And those might be moods that you have that vary between the day, but personality traits are stable. They describe things that you see in somebody who's 10 years old, and then when you meet them again 20 years later, they're still basically kind of the same person. So in humans, the Big Five model that I just talked about is the way that this tends to be organized by the literature. And they're interesting for a variety of perspectives, but one of them is that personality traits are very good predictors of many important life outcomes. Things like educational attainment, how far you make it through school. Do you finish high school? Do you finish university? Do you, whatever. As well as lifetime success, measured however you define success, I suppose. That's a pretty subjective decision, honestly. Or even longevity, how long you live. And that's all well and good. I think this sort of describes the value of understanding personality, because clearly it has an effect on things in your life. But another question that maybe is more relevant to us as geneticists is this relationship that's been observed between personality and apparent reproductive success. So this has been noted many times. Hold on a second. Let me just go to the chat window here. Nathalie asks, just an aside, has there been any effort to possibly redefine the introversion descriptions? I'm also an introvert, so I feel slightly attacked. Yeah, I don't know. I mean, like, many of these personality traits, the Big Five, there's clearly like a good side and a bad side to be on. So, for example, if you go back to agreeableness, obviously it's better to be agreeable than to not be agreeable. Society probably needs a couple of people out there who are disagreeable in order to make things happen. We wouldn't be anywhere if, America wouldn't be anywhere if George Washington and his friends had been agreeable. America probably also wouldn't be anywhere if Abraham Lincoln had been agreeable and had just said, let's go with the flow. I'm only thinking of America because it's hard to think of history under pressure. But anyway, you need some, but generally speaking, we think agreeableness is a desirable trait. We want our kids to be agreeable. Conscientiousness is the same way. Clearly, it's better to be conscientious than not by our standards. even though people can be all over the spectrum on that. And extroversion and openness too. I think openness is maybe a bit more subjective, but I think you're not really saying something nice about a person if you say they're not open to new experiences. That's not a great thing. So extroversion is sort of value laden in the same way. By reference or by analogy to the other scales, extroversion is clearly something that's good, and then the converse of that, introversion, would be the bad thing, which is not. I think in that particular area, there's been a lot of effort in sort of the psychological and popular psychological literature to destigmatize introversion, because clearly it's not, being introverted is not nearly as bad as being disagreeable or unconscientious. So it's that particular scale should be taken as more value neutral. I remember when I was in undergrad, I read a book about introversion, and it sort of changed my life in a way, because it kind of helped me understand my personality. Because before that, I thought I was disabled somehow or had some kind of big problem. But it turns out that I'm just, on a certain spectrum as far as personality traits go. And that really, if you learn about that kind of stuff, it can really help a lot of questions about your life lock into place. So it's worth considering. Oh, and then neuroticism is the only one of these five traits that's kind of the reverse, where high levels of neuroticism are not good, but low levels of neuroticism are good. So high neuroticism is not ideal, and we'll see that play out in a couple slides from now. But anyway, so there's this relationship between personality and reproductive success, where here Berg and all are publishing the relationship between number of children, grandchildren, and everything else, and personality traits. So numbers above 0 mean that the trait is positively correlated, and numbers below 0 mean the trait is negatively correlated. And the strongest thing we see is that people who are high on extroversion tend to have more children, and people who are high on openness to experience and conscientiousness tend to have fewer children, which is a rather interesting concept. And this has been replicated quite widely. This is just a table showing the times that this has been tested, and Berg et al. Has the largest study, and thus we have the most confidence in it. But it does seem as though the traits do affect your reproductive success. In spite of this, investigations of the evolutionary pressures seem to suggest that these traits are under balancing selection, meaning that while there's variation, there's not a great deal of pressure in one direction or the other to drive natural selections. Because you'd think if extroversion was associated really with more success all over the place, why would there be any introverts around? Because all of the introverts would have been outcompeted and bred out of existence by all the extroverts. But it turns out that we actually function pretty well when there's a balance between the two types. Go figure, we're all important. So twin studies and family studies and all those kinds of things suggest about a 50% heritability for personality traits. And as far as your individual traits go, the single greatest predictor of your score on a given trait, extroversion, neuroticism, etc., is your parents' score. So there's clearly a heritability aspect here. But I think that at least as far as the relationship between personality and reproductive success is concerned, it seems to be culturally or temporally dependent. So here is a paper from Jokela et al. In 2012. No, it's just Jokela. It's nobody else. It's just him or her. I can't remember. And here what they're measuring is the relationship between personality scores and reproductive success. But they're looking at different birth cohorts. People born in the 20s, 30s, 40s, 50s, and 60s. Or sorry, those are the traits. 20s, 30s, 40s, 50s, and 60s. So the different colored bars represent people who were born in different decades. Okay? And then the y-axis represents their odds of, or sort of, essentially the relationship between that trait and their reproductive success. So a positive value means that they're having more kids, and a negative value means they're having less kids relative to how extroverted they are. So here in men, there's no effect of extroversion in this scale at any of the time points measured, and there's really no effect of anything else in men. However, there is an effect of openness to experience. Right? Well, yeah, there isn't actually an effect of extroversion, but it doesn't change over time. There's an effect of openness to experience such that it has a in people in sort of boomer era people born in the 60s, the more open you are, on average, the fewer kids you have. And the same is true in women as well. The more openness to experience a woman has, the fewer children she'll have over her lifetime, but this effect becomes stronger in the 60s compared to the 20s. And women are also have a sex-specific effect of conscientiousness, such that the more conscientious the woman is, the fewer children she tends to have, but only in this sort of later boomer, 1960s and beyond. So this is kind of an interesting thing. I guess what it would imply is the more open you are to new experiences, the less likely you are to have kids, maybe because you're too busy traveling the world and enjoying your income and not having babies take up all of your time. Who knows? Having a kid is also a new experience, so you should be open to that as well, but it's hard to say. I mean, I guess, strictly speaking, pretty much everyone on Earth is going to have kids at some point. The number is over 95%, I believe, is going to eventually become a parent. So we're really talking about differences of degree, not so much qualitative differences. But I think it is important to take a look at the way personality differences manifest differently depending on our society. Clearly, there were societal changes between the 20s and the 60s in terms of the role of women, what personality traits were desirable, the way that courtship worked, the idea of what the family structure should be, and many, many other things. And this interaction between personality and society is what drives this reproductive success. So personalities really have different effects depending on which society you happen to live in. You may have a personality that would work very well in one place, but work terribly in a different place. So that's always something to consider, by the way. If you're finding you don't fit in somewhere, whether it's like you don't fit in at Carleton because you're not extroverted enough, or if you don't fit in at your job because you're too extroverted and you keep screaming at the customers, or whatever the case may be, oftentimes it's not really a problem with you or anything else. It just happens to be a poor fit between your personality and wherever it is you happen to be. If you're in the wrong time, like if you're from, if you're like a 1920s guy and you're really annoyed that you can't you can't act like a 1920s guy and dress in a zoot suit and tip your hat all the time, then I don't know what to tell you. But if it's something you can change, it's worth considering. Now, if you think back to our behavioral genetics lecture, I talked about how the heritability of traits seems to vary over the lifespan, such that the twin studies estimate the heritability of intelligence, or cognitive ability as they call it, is fairly low when you're young because there's not much intelligence there to measure, but becomes relatively high by the time you're about 10 years old. And accordingly, the contribution of the shared environment between twins decreases. Personality is similar. The heritability of personality is very, very high when you're young, and it gradually decreases, and the environment tends to have a stronger influence, although it never really decreases that much. So if you're looking at the influence of genetics and personality, the influence will be stronger when you're young, presumably because you're more under the control of your parents and you're able to, you absorb more of your behaviors in general from your parents. But as you age and as you enter into the world, the environment you have begins to take on a stronger influence. So these are kind of rather contrasting if you think about this relationship between heritability of cognitive ability and heritability of personality. I guess, what I'm getting at here is that it depends, it matters when you measure these things in the lifespan. So we have some genetic data as well, and as is frequently the case in this class, a number of candidate genes were studied early on for their potential relationship to personality, and these would include DRD4, the 5-HTT-LPR polymorphism on the serotonin reuptake transporter, as well as COMT, the catechol O-methyltransferase, whatever it is, the VALMET polymorphism on that one that we talked about back in the behavioral genetics course, or lecture rather. These were all exciting at first, but as we've seen before, they did not stand up to genome-wide scrutiny, meaning, as always, you really have to be quite skeptical of candidate gene-based methods. And in the meantime, GWAS studies have gradually racked up trait-associated variants, but it's been a slow process. Neuroticism is one of the 5 traits that has the largest number of SNPs. So this is what this is showing here. This is just showing the results of GWAS studies over time. The number of significant SNPs or GWAS hits is plotted on the y-axis, and the year the study was done is plotted on the x-axis. So you can see that the highest number of SNPs is found for neuroticism, That's in this recent 2017 study. Neuroticism is also the highest in the 2016 era of studies. For whatever reason, neuroticism seems to be especially amenable to GWAS investigation. Does that mean it's a unique kind of personality trait? It's a little difficult to tell. But anyway, it does seem to have a lot of SNPs that are linked to it. So as far as the GWAS studies go, I'll tell you about some of the more recent ones and their findings. In 2017, Lowe et al. Published a large meta-analysis of personality-associated variants, and like many people do nowadays, they use data from 23andMe, which is that company where you can send them your DNA and they'll tell you what your eye color is and about your risk for different kinds of things. They're kind of a privatized version of the public the public research we do in genetics involving polygenic risk scores and so forth. But they have a vast quantity of data, and because everybody who submits their DNA there is phenotyped by online quizzes and surveys, you can pull that data out and combine it with academically obtained data and get these gigantic data sets for doing genome-wide association studies. So depending on which trait they were looking at and how good the phenotyping data was, the authors of this paper were looking at between 123,000 and 260,000 participants. So these are very, very big numbers of traits. They dwarf, by the way, the genetics of language studies that have been done so far. So I've posted up the Manhattan plots for each of the 5 traits. They're a little bit small here, but you should be able to find the paper if you want to look at them more closely. The authors didn't find any SNPs for agreeableness or openness. They had some ones that were pretty close. You can see this line here is the p-value cutoff for genome-wide significance. And they had some areas that were definitely like getting close, but they didn't quite make the cut. But they found one new one for neuroticism, or sorry, 2 for neuroticism. And most of them they found for extroversion for whatever reason. So they found a total of 8 SNPs altogether, which is a pretty limited haul if you think about the fact that they studied this many participants. But this is a common, complaint about GWAS studies. And most of the SNPs they found were linked to extroversion. And I should point out that 6 of the SNPs, 6 of the 8 that they found, were also found to be EQTLs, or expression quantitative trait loci in the brain, by the GTEx study and other previous works. So this is telling us that what's happening is taking place at the level of the brain, and these SNPs actually affect the expression of genes in the brain. And consistent with this, Luciano and company back in 2018 studied neuroticism specifically. They were just focusing on it because it has so many documented SNPs, and looked at the gene ontology terms that are involved there. And if you, of course, you remember gene ontology from, I guess, your second assignment, and they're doing the same basic thing. They're just taking lists of genes that have neuroticism-associated SNPs, and they're submitting to tools like WebGestalt or whatever, there's a lot of different gene ontology tools you could use, and they're just asking what terms come up. And what you see is neuron spine, homophilic cell adhesion, neuronal differentiation, more cell adhesion stuff, and neurogenesis. Cell adhesion, I should say, in the context of brain development, mostly has to do with like protocadherin genes and other kinds of cell adhesion molecules that help synapses find where they belong and stick together. There's really quite an interesting story to tell about those protocadherins, but I guess I don't really have time for it. Anyway, so cell adhesion molecules in the context of brain-related phenotypes usually refer to the process of brain, axonal and synaptic development. These processes from cells finding each other and sticking together and forming connections. So it's not surprising, I suppose, if you look at the genes that are linked to personality and you find that they all have to do with the brain somehow. What else would they be? Wouldn't it be funny, folks, if you did this study and found out that all of the genes annotated to the heart? I think that'd be, it'd be rather romantic, but it would be very confusing to explain scientifically. But, once again, we're coming to the conclusion that, by very indirect means, that the brain is responsible for personality. And I was thinking about, I think I brought this up kind of on a whim, talking about, Hippocrates and Aristotle and these kind of old-fashioned philosophers who weren't really even sure where the mind was located. Was it the heart? Was it the liver? Was it the brain? Who knows? Aristotle didn't even believe, Aristotle thought the brain was essentially like a radiator for the body. It didn't have anything to do with your thoughts. I was thinking about how far we've come as a scientific enterprise. So I'd like you, for those of you playing along at home, to just, maybe pause this video or just take a moment to think about this. Think about what I'm talking about here. These are gene ontology terms that have that come from genes in which there are neuroticism-associated SNPs. And think about all of the different things that are involved in this piece of information. Think about the DNA. Think about what a SNP is. Think about genome-wide studies. Think about gene ontology. Think about transcriptomics that figures out where genes are expressed. Think about all of the experimental work that goes into figuring out what, if a given gene has to do with neuron spines or differentiation. Think about all that stuff, and then how, after it's all been done, you can go back and look at population genetic variants and link it all back together. And think about how complicated it is to be able to understand something like that. And the fact that hopefully, if you've gone through this class and if you are combining it with other things you've learned in neuroscience, you probably do understand it to a fairly high degree. So, sometimes you just have to kind of appreciate what it is you understand and know and how meaningful that is and how much it differs from what you would have known just a few years ago and what the smartest people in the entire world would have known a few thousand years ago. It's just something to think about if you're in the mood to reflect on it. So we have genetic correlations as well, and I think in the case of personality, this is fairly interesting because, it's a real curiosity whether personality traits correlate with other things. People have brought up before the idea that psychological disorders many times could just be extreme versions of normal human personalities. So, or extreme manifestations of different normal aspects of human variation. So you all remember how to read a correlogram. I don't need to go over that again. So you can see this correlogram has been clustered. The traits, agreeableness, conscientiousness, and extroversion all cluster together. They're all positively correlated with each other. Neuroticism is negatively correlated with all of them. This is because neuroticism is the only trait where high numbers are like bad, or the higher you are in the scale of neuroticism, the worse it is, technically, whatever that means. Openness to experience has kind of more of a mixed set of correlations with everything else. You also see a cluster of positive correlations with the different kinds of mental disorders they studied. Schizophrenia, bipolar disorder, and major depression all cluster together, again suggesting that there's some kind of underlying genetic architecture to these things. And there's many, many other relationships as well. Surprisingly, openness is not genetically correlated with depression, which is interesting. Let's see here. Openness to experience. Depression is so there's not a significant correlation with depression, which is kind of surprising if you think about it, I guess. Or sorry, it's extroversion. Extroversion is not extroversion olive oil. Extroversion is not correlated with depression to a significant degree. Let's see if we can find that actual. Yeah, so here it is. Extroversion. They really could have actually put names here instead of these stupid numbers. It makes it very troublesome to read. But anyway, extroversion is the third trait from the top. And major depression is here. So the correlation between extroversion and depression is 0.02, which is not a correlation at all. So there's no relationship to that, meaning that you can be just as happy whether you're introverted or extroverted, I suppose. You just have to learn what place in the world is best for you. Schizophrenia is correlated quite strongly with openness to experience, which I find rather interesting. That correlation is right here, I believe. Openness is 1, 2, 3, 4, 5. Yeah, so the correlation between schizophrenia and openness to experience is here, 0.36. That's actually one of the highest correlations on the map. So that's kind of an interesting concept to think that maybe schizophrenia is a very extreme version of openness to experience, where you're so open to every experience that you begin to be open to things that aren't even there in the world. That's a bit fanciful, but you never know. It's something to think about. And then you can also take the genetics of personality traits and mental illnesses and cluster them together using principal component analysis and hierarchical clustering. Don't worry exactly how this was done, but essentially what they're showing is the kind of hierarchical relationship between genetic signals for these traits. I realize I'm running a bit long here, but I'm just going to keep going. So this clustering pattern shows you, basically, that genetically speaking, certain personality traits are very similar to certain mental illnesses. So for example, extroversion clusters with ADHD. Now, does that mean that ADHD is sort of genetically speaking an extreme version of extroversion? Does that sound right to anybody? I don't know. Agreeableness and conscientiousness cluster together. They're very similar traits if you think about it. Anorexia nervosa is a little bit on its own, but it does cluster somewhat with neuroticism and major depression. I've heard about the link between neuroticism and risk of anorexia nervosa. So that's, on the basis of just psychological research, I think that's plausible. It's interesting that the genetics also seem to show that. ASD is separate from other ones, but it's sort of in the same superfamily as schizophrenia, bipolar. Those two are very close to each other, and openness to experience. So I keep showing these things because I think it's very interesting. These geneticists are not psychologists. They're people who would not be able to design a psychological assay or conduct a structured interview for the most part, or any of the other kinds of things that would help you diagnose or get to the bottom of any of these phenotypes. They're approaching it in a way that's fairly naive to the existing theories of psychology. And yet, when you take this data and naively cluster it out and just let the things group together where they're similar, you get these links or correlations between different traits, different phenotypes and different illnesses that fit the psychological profile that people have thought of very nicely. So these sources of evidence really do seem to converge on a similar underlying neurodevelopmental genetic structure for many aspects of personality and mental illness. So in summary, GWAS studies remain limited in their ability to detect weak effects, low-frequency variants, as well as dominance and epistatic effects. So, of course, we only have 8 SNPs really that we're working with from the biggest study, and when you put everything together, you have maybe a few hundred that are kind of somewhere near significant and workable as a model. So there's lots of missing causality to go around. They could be due to epigenetics or due to these other kinds of effects. The existence of genetic correlations between personality traits and psychological disorders seems to point to, as I've said many times, a larger genetic architecture of inter-individual variation. Rather interesting to consider the possibility that that's the case. It also, I think, points you to where to look for molecular biology of these conditions. Much more often, we see neurodevelopmental genes involved with these things, not genes involved in like monoamine neurotransmission or whatever else. It seems to suggest that neurodevelopment is really where you've got to be looking if you want to understand both personality and mental illness. And I think neurodevelopment has been a little bit underemphasized in the mental illness space, I should say, because none of our drugs, obviously, none of our treatments can really do anything about development. So we don't tend to emphasize that as a cause because it's kind of in the past. There really is not a lot that can be done. It's also possible to combine personality GWAS data with transcriptome data from drug studies. And so I just wanted to point this out that these GWAS studies may also point to better treatments because they tell you what genes are differentially expressed and differently involved in phenotype, and thus and you can combine that with knowledge of which genes are affected by drugs. You can actually find that in the UCSC genome browser. If you look at a gene, it'll tell you what drugs affect that gene. So it's possible to put all this together and potentially develop better treatments for psychiatric disorders. But ultimately, we're faced with the reality that the path from GWAS to biology is not an easy one. Whether you're talking about language or personality, getting from genome-wide data to the actual mechanics of personality, like how is it that having a difference in your dendritic spines makes you more likely to jump out of an airplane? It's a long way to go to get from that basic aspect of biology all the way to the complicated behaviors we show in our everyday lives. And I wouldn't begin to claim that we're anywhere close to that. But the good news is, if you're in this field, you'll have job security because we're not going to figure this stuff out anytime soon. But I think, in the next 10, 20 years, we're going to see some really great stuff. So there's a lot to look forward to. Anyway, folks, sorry I went a bit long today, but there's just, there's so much to talk about and the year's coming to an end. So hopefully you all learned something and found this to be interesting. Remember that your assignments are due, I think, in about a week. I've heard that there are some problems with the Great website. I think that's been fixed now. I checked Great this morning and it looks like it's working. So to those of you who have contacted me, try again, please. And keep on it. I would really suggest getting this done sooner rather than later, because if the websites aren't working for you and it's the night before the deadline, we're going to have an awkward discussion, I guess. I don't really know what I'll say, but it's just much easier if it's done, and then we don't have to even get into it. So thanks to everybody, and we'll see you next week. I'll have some information maybe next week about what the final exam is going to look like and what you should focus on for your studying. Thanks, everyone, and we'll catch you next time.